Okay, so we're going to talk about the cell membrane today. I'm going to give you the basics of the membrane in this video, and then in other videos we'll get a little bit more specific. But hopefully you have the picture of the membrane drawn. If you don't, you might want to take a second and draw the membrane so that you already have it done, so we're not spending time doing that. But the membrane you have has a lot of different purple blobs all embedded in it. And everything you see in your image that you have drawn there that's purple would be considered a protein. Then within the membrane where these proteins are embedded, you have these little guys, okay? And what I'm drawing right now are all the heads of what molecule? And that molecule is the phospholipids. And these phospholipids have heads, and then each little phospholipid head has two hydrophobic tails attached to it. So these little guys are what keeps the cell from falling apart. Which organelle is responsible for creating, these phospholipids whenever any organelle needs a new membrane? Okay, they are created by the smoothie R, and that's the endoplasmic reticulum. Now, this is a phospholipid bilayer, and we typically think of it being around the complete cell, which is absolutely true, but so we can say it surrounds the, surrounds the cell. All right, it can surround the nucleus. It surrounds certain organelles like your mitochondria. It can surround the chloroplast, your lysosomes, your vacuoles, anything that is packaged up. Okay, this is what we package it with. We package it with these membranes, and these membranes are called a phospholipid, okay, because literally that's what they're composed of, and then they call them bilayers. And that's because if you look at them, there's two of them in every layer. These phospholipids orient themselves this way. All right. And the reason the tails are all towards the center is because all the tails, they're all kind of kinky, like looking, they're all bent like this. These tails, this region is hydrophobic. And hydrophobic means it does not like water. Now, the heads of your lipid, your phospholipid, are hydrophilic. And that means they like water. And so we're going to draw it with a little smiley face. So the tails all want to hang out together because they all hate water. And the heads all like water. Now, when you were drawing your membrane, you should have labeled in our image, and it could go both ways, but in our image, this side is going to be the cytoplasmic side, okay? So this is the inside of the cell. And if we were to, actually back up and look at a cell and then draw the entire thing, this little piece would only be a small section. This whole thing goes all the way around the cell, and all throughout all those phospholipids, all along the membrane, you have different proteins all embedded, okay? And the proteins are floating around almost like little kids in the wave pool, and they're on those floats and they're bobbing up and down. So that's how you can imagine these. These are all the little people in the wave pool, and these are their big floats. And it's moving and it's fluid. And so because of that, we call the membrane fluid mosaic, okay? Fluid means to flow and move. So this is a dynamic process because it's exposed to water and it's floating around, and these are little fat molecules, so they're kind of greasy and slippery. And then mosaic comes from the art term where you have images where people make pictures with little tiny pieces all put together. And that's coming from all these different proteins that are embedded in this membrane and constantly moving and flowing around. So if you look at a cell, 1 minute, you could see all these proteins on one side, and then they'll flow and move to another side. Okay, so the membrane is not solid, it is not dynamic, it's constantly flowing and moving. All right, so with this hydrophilic head and hydrophobic tail, if you were to look at this little layer, the outside, which is extracellular, okay, is hydrophilic because all out here is water. And if you remember, your body is composed of about 70% water, and then other organisms can be as much as 90%. So this is happening in all organisms that have membranes. Then on this side, on the cytoplasmic side, that fluid, that liquid cytosol, is mostly water. It's that consistency of an egg white. And so it's also exposed to water. So these guys are good with being, exposed to the inside of the cell. And these guys are good with being exposed to the outside. It's this middle chunk right here that makes this cell waterproof. You are not getting through this membrane if you're water. You cannot just come in and out of the membrane. And so this is what keeps and maintains the integrity of this cell. So we're going to put that up because that's a big. So the membrane maintains the integrity of the cell. And that just means it keeps things out that should stay out, and it allows things to come in that it wants to come in. But these phospholipids, they make it waterproof, and so the only way you're going to get in is if you're allowed to by them, and there's a special situation for that, or if you're allowed to pass through these proteins. Okay, so the membrane needs to stay very fluid, but sometimes organisms are exposed to extreme temperatures, either on the very, very hot side or on the very cold side. And so since these membranes are made of fats, if it was really, really cold, fats can solidify. And if your membrane was to become solid, then your cells would become too rigid and things couldn't get in and out and the cells would die. So when people have frostbite and things like that, it is a consequence of water inside their cells freezing and their membranes just becoming too solid. On the other end of the spectrum, if it's too hot, so we've got too cold, that's no good. And then we have too hot. Okay, that's also no good because fats, when things get hot, they move a lot faster and they get even more slippery. So since these are basically little fat molecules, when it's too hot, they would slip and slide around, and then your cell might actually fall apart. So embedded in the membrane, and you have it drawn on your image, and it looks like four rings, which 4 rings represents a steroid. So it's like, I think it's 3 hexagons and a pentagon. That's going to be, so ring, 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 and ring. So this would be a hexagon, this would be a hexagon, this would be a hexagon, this would be a pentagon. And this molecule embedded between these phospholipids is called cholesterol. Okay, and cholesterol is a steroid, and its main function is to maintain the fluidity. So when it's too hot, the cholesterol will hang on to all the phospholipids and keep them from getting too crazy and slipping away and the cell falling apart. When it's too cold, it changes its purpose and it continues to allow for movement, and it keeps the membrane from solidifying. Now, there are temperatures where it just really is way too cold or way too hot for this cholesterol to do its job, but in general, it keeps your membrane fluid, and so therefore everything's flowing and moving, and then your proteins can start doing their job. Okay, now with the proteins, if you notice in your picture, you're going to have some that go all the way through the membrane, and then you're just going to have some that are like just attached to the inside of the membrane, and then some you might have that are just attached to the surface, the extracellular side. Okay, so if they go all the way through the membrane, so that there's a part of them that's on the outside of the cell and a part of them that all comes all the way through to the inside, you can consider them passageways or communicators that are talking to something on the outside and inside, or vice versa. Or they can be where things pass through. So these that move all the way through the membrane, they are called integral. So your integral proteins move all the way, or they span the entire membrane. So there's a portion of them that's on the surface, and there's a portion of them that's on the inside. They are going to be doing different functions than these proteins that are just attached to the extracellular side or the intracellular side. These guys are called peripheral. And periphery means like on the edge. So peripheral proteins do not go all the way through the membrane. So since this protein right here doesn't quite make it to the top, he's still considered peripheral. This guy right here is also peripheral. But these that move all the way top to bottom are called integral. Now, an integral protein, just going to show you a little bit, because he's going to be embedded within the membrane. So he would be side by side with these phospholipids. This guy is hydrophilic, and then we have phobic, and then we have phobic, and then we have philic. Okay? So they love water, hate water, love water. And they can mix with water, can't mix with water, can't mix with water, can mix with water. So as you move inside or outside of a cell, that's what's going to happen to the appreciation or interaction with water. So proteins that are integral have that same type of concept. Their top part is philic, and their bottom part is hydrophilic. But their middle part is hydrophobic. And that's the way they are able to stay embedded in the membrane. So it's hard for them to leave and get out of the membrane because this part wants to stay in the middle, and these parts are happy where they are. Now, if we looked at a true protein, this is an artist drawing to make you feel like you understand the structure of it. It's this big purple blob inside your cell. It's not actually a purple blob, but that's for effect. But the way an actual protein looks is, if we remember from the unit where we talked about protein structure, proteins either twist into an alpha helix or they bend into a beta pleated sheet. So these proteins at the molecular level actually are alpha helix that twist 1, 2, 3, 4, 5, 6, and then we make our final pass 7. Okay, so with some little loops up here on the top that are a little bit bigger, and some loops are on the bottom. So this protein right here can sometimes be drawn the way it actually structurally looks as a loop, an alpha helix that's looped 1, 2, 3, 4, 5, 6, 7. Okay, so with one piece hanging out at the top or the extracellular side, and one little piece that's looped down into the intracellular side, the cytoplasmic side. So this is the actual structure, and you will see it drawn that way in some of the membranes on some of the pictures. They call this extracellular side, they call this the N-terminus. Okay, now terminus or terminal means the ending of something. So that's your N-terminus, and then this side is the C-terminus. And that's just to give you direction. So the C-terminus side of the protein chain, because that's one long chain, is the one that's inside the cytoplasm. That's how I remember it. And then the top part, the N-terminus, is the one that's on the extracellular side. Okay, so now that you kind of have a little bit more better understanding of how the proteins look, and that you have integral proteins and you have peripheral proteins, I do want to show you the 6 main categories of proteins that we would find in the membrane. Because your membrane is just going to be all these different proteins that are embedded in it. And depending on the type of cell and the function of the cell, it's going to have lots of different types of proteins. So it's not just this is exactly the same as this. They're both proteins, but they may allow different things to happen. So in your book, you have this page. Let's see if it'll focus. Awesome. Okay, so it's not going to focus. All right, so I will have to draw it for you. Okay, so I had to get it focused. It wasn't quite working right. Okay, so with your proteins, there are 6 main types. Okay, so within these types, there's lots of different examples. But your 6 main types, your first one is transport. Okay, and those are going to be those proteins that allow things to pass in and out of the cell. They can be active or they can be passive. Okay, active means they require ATP, and passive means they don't. So that's going to be your osmosis, your diffusion, things like that. Second type is going to be enzymatic proteins. Okay, and enzymes speed up chemical reactions. So their whole purpose is to create chemical reactions on the surface of the cell. See, and these are definitely going to be integral, because if you want things to pass in and out of the cell, it's going to have to go from inside to outside or vice versa. Over here, these are also going to be integral, because they're going to allow things to reactions to occur, and it's going to cause inside to outside breaking down things or making things. All right, your third category of transport protein of membrane, membrane proteins. Okay, so your third category of membrane proteins are ones that do signal transduction. Okay, and their job is to send, receive, and respond to chemical signals. So this is how you get your cells to do what you need them to do. Hormones can be chemical signals. Different medicines can be chemical signals. Your body can send out chemical signals. Your immune system sends out chemical signals. Signals are basically just any type of molecule that can be received by a cell, and then when the cell receives it, it responds. If a cell does not receive the signal, there is no response. And if a cell does not have the protein that does the receiving, then there is no response. So not all cells have the exact same signal transduction pathway proteins, but these are also going to be integral. So basically, it would be like your cell phone. Like you get a message, you receive it, and then you can respond to it. All right. The fourth category of membrane proteins is cell-to-cell recognition. And these are extremely important because this is how cells understand or know that another cell is a good guy. Okay, and these are based on glycolipids and glycoproteins on the surface of the cell. So that's going to be extracellular. So therefore, since it's only on the surface, these are going to be peripheral proteins. And basically, they're only on the extracellular side. There's no need for them to be on the intracellular side. So I'm going to go back to the picture of the membrane. And a lot of you, when you drew it, you had these little weird green, on some of these, you had these little green pieces coming off. And they called them glycolipids if they were coming off of one of our phospholipids. And then if they were attached, oh, shouldn't draw them there, I'll draw them to this one. If they were attached to a protein on the periphery, then they called them glycoproteins. So this is a glycolipid because it's a glycogen molecule attached to a lipid. And then this is a glycoprotein because it's a glycogen molecule attached to the peripheral. Now, the job of these, okay, so I'm going to go back to this page. We call these things, they're like identification tags, okay, or flags. So they're flying on the surface of your cells, and they identify your cells as being the good guys. So every cell in your body has the exact same flags. And this is how your immune system can recognize that your cell is supposed to be there, it's a good guy, and it doesn't kill it. So in cell-to-cell recognition, all your cells have to have the same flags, and the reason they have to have the same flags is so your immune system does not kill them. This is also how we do organ donation. When we're trying to find organ matches, we're actually matching these glycolipids and glycoproteins because people who have the same flags, then their body will recognize that organ and won't kill it. And so sometimes if you have some sort of issue and your cell isn't making the right flags, then your body starts attacking yourself and you have something called an autoimmune disease. All right, we'll get more into those type of proteins later. All right, so far of our 6 main types, we have transport, enzymatic activity, signal transduction, so they send and receive signals, and then cell-to-cell recognition, whereas these flags on the surface of the cell let your whole body know that they're the good guys. All right, and then the last two are, E is going to be intercellular joining. Okay, and then F is going to be attachment to the extracellular matrix. That means the outside or the intracellular microskeleton. Okay, so these proteins are, if they're intercellular joining, they are allowing cells to attach to adjacent cells. Adjacent means beside. Okay, so if you see this cell and it needs to attach to this cell, they're going to have proteins on their surface that allow them to stick together. Some cells, they have a lot of them and they stick very tightly together. And some cells, they have little spaces between them. But whenever two cells are sticking together, because two cells stuck together, and then you have another one, a group of cells that are working together to perform a function, so you get thousands and millions of these, is called tissue. Then when you get all the tissues sticking together, they call it an organ. So you need all your cells to stick together because very, very few of them just flow around and aren't attached. So some of your blood cells don't have these because they don't stick to anything and you don't want them to. And then some of your immune cells are missing these proteins because we do not need them to stick to anything. Some of the immune cells, the only proteins they have stick to the virus or the bacteria that they're looking for, and so that's the only thing they will attach to. These are long lasting. Okay, so once they stick, they stay that way. That's an S. All right, and then The last type is the attachment to extracellular matrix and or the intracellular. So those are going to, if they're extracellular, they're just going to be on the periphery on the outside. Intracellular, they're going to be on the inside. So these guys are cells to cells. This is attaching to a ligament or a tendon or something that is not the same type of cell. So this would be very similar to what we call cohesion with water sticking to itself. And this would be what we would compare to adhesion, where water was sticking to substances that were not other water molecules. So this holds the cells together, and this holds the cells in place. And then on the inside of the cell, if you were to look at it, it's all these little tiny filaments and tubules that are very similar to muscle fibers. And it keeps the cell in a round sphere instead of it collapsing and being too flat. Okay, so I just want to add one more thing over here with your cell-to-cell recognition. A lot of people ask me about blood types. So you have type A, type B, type AB, and type O, and we'll get way more into detail in that in another chapter. But this is how the different types come about. There's two different flags. There are flags for the A, and there are flags for the B. So if you were type A, you're flying the A flags. If you're type B, you're flying the B flags. If you're AB, that means you're one of those weird ones that have two flags. You're cheering for both teams. And then type O, no flags, okay? And so your body recognizes these based on, do they match you? This guy doesn't stress anybody out. So that's why people who have type O blood can be donors, because they can give blood because their flags don't offend anyone. Type A can only be given to type A or type AB because they carry that type A flag. Type B can only be given to type B people or to AB people because they both have the B flag. AB it can donate only to ABs, all right? But it can receive all types of blood because you can give it type A, you can give it type B, you can give it type O, or you could give it type AB. So it's all about these flags matching and being recognized as safe. All right, so that's the end of the video. So you're gonna have some questions now. And I'll have another video next about some of these more specific types of movement and transport. But that is your introduction to the phospholipid bilayer.